Okay, how's it going, everybody? In this video, I would like to talk about the chi-square test. And in genetics, we care about the chi-square test, at least at this part in our course, because we are studying ratios, genotypic and phenotypic ratios that result from doing genetic crosses, right? So we talked about Mendel's ratios of 3:1 and let's say 9:3:1. So those are his phenotypic ratios of the monohybrid and dihybrid cross. And it's important to remember that, or note that these ratios are, they don't have to be exact, and because of this phenomenon called chance deviation, that, we'd be very surprised if the ratios were exactly 3:1 and 9:3:1. So what is chance deviation, or when does it occur in our, let's say, genetic experiments? And one place it can occur is during fertilization, right? So for if we were looking at, so this cross here, where we have two tall plants heterozygous for this pair of alleles right here, this plant makes a big D gamete and a little D gamete. This plant makes big D gamete, little D gamete. So the process of these coming together, these gametes, to make the zygotes, involves some level of chance. And as a result, we would expect, especially when we're looking at small numbers of offspring, for our ratios to deviate from 3:1 and let's say 9:3:1, or other ratios, which we will see coming up in the next lecture. So we have a way to know how much deviation is abnormal, how much deviation from an expected ratio is unusual, and that's the chi-square test. So let me write that down here. Chi-square test. So, okay, so for example, let's take, let's say we're studying a new trait. My pencil is breaking here. Okay, it's okay for now. So a new trait in P, say we discovered a mutation that causes small p size. So this is even smaller than the normal size of p, which is what we will call large. So, and our hypothesis, let's say, is that the small p trait is caused by a homorecessive genotype for this gene that we will symbolize with the letter S. So two small s's causes the small p phenotype, and at least one big S allows for the large p size. So we can predict what would happen if we were to cross, say, true breeding plants that only made large p's with true breeding plants that only made small p's. If we were to cross those, that was the P1 and the F1, all the plants should be heterozygous, all from a large P. We grow these up, and their offspring self-cross here, or cross to another individual from the F1 generation. We would expect 1/4 and 2/4, 1/4 to be homozygous dominant, 2/4 to be heterozygous, and 1/4 homozygous recessive. So 3/4 tall, large, and 1/4 small. So the ratio is 3:1. So if our hypothesis that this new trait is caused by the homozygous recessive condition is true, we do this experiment, we're expecting a 3-to-1 ratio of large to small in the F2 generation. Let's say we perform the experiment, but in the F2, we see a 4-to-1. Let's say we only have a small sample of offspring. So 80 large P, 20 small p. So we need to know if this 4-to-1 ratio is so different from the 3-to-1, deviates so much from 3-to-1 that our hypothesis, cannot be true. So we need to know if this deviation is significant, because if it's significant, then we need to go back and we need to revisit our hypothesis. Maybe something is wrong with our hypothesis. Maybe the small p trait is caused by mutations in multiple genes. Or there could be another explanation. However, if this deviation is not unusual, given this sample size right here of 100 offspring, if this is not unusual, if it could be attributed to chance deviation or chance events, say during fertilization, well, then our hypothesis could be true. And our data here supports this hypothesis. So essentially that's what the chi-square test does. It allows us to determine how much deviation is normal from a prediction. Well, how much deviation is normal in an experiment that involves random events. So before we look at our genetic example, let's look at a coin toss example. I like coin toss examples because they're relatively easy to understand. So let's say we flip a coin 10 times. Now we would expect 5 heads and 5 tails for a 1-to-1 ratio. Now, what happens if we do the experiment, flip a coin 10 times, and we observe 6 heads and 4 tails? So that's not the 1-to-1 ratio we expected, but it's not too different, right, from 1-to-1. I predict that this is likely. This is, not too unusual. So we wouldn't change our hypothesis that, every time we flip a coin, half the time it should be heads, half the time it should be tails. I wouldn't change the hypothesis that you flip a coin many, many times, the ratio of heads to tails should be 1-to-1. However, what if we did the experiment? Let's say this is experiment 1. This is another experiment, experiment 2, say maybe a different coin. We get 10 heads and 0 tails. Well, that would be, to me, that seems highly unusual. It doesn't seem impossible, but it seems highly unlikely, and it would suggest, it's so unlikely that, maybe something is wrong with the coin. But we don't know. We need a way to know how unlikely is this? How unlikely? And how unlikely is this? I'm having trouble spelling unlikely today. So, okay, we need to figure this out. And the chi-square test is going to tell us, how unlikely this outcome is and how unlikely this outcome is. So how does the chi-square test work? Well, it's relatively easy to implement. So there is an equation that you won't need to remember because I will always give this equation on a test or a quiz. But you need to know how to use it. So O is for observed, E is for expected. You're going to square that and you're going to divide it by the expected. This thing right here means you need to sum up this value for the different categories. So in our experiment here, we have a couple different categories, right? We have 2 categories total. We have heads and we have tails. So I could put together a little table here. Let's see. So we had a total of 10 tosses, right? 6 of them were heads in the first experiment, right? And 4 were tails. We expected, let's say this was the observed, we expected 5 heads and 5 tails. So now we need to use this equation with these numbers here. So for the heads category, we're going to, this equation here, we're going to use it for the heads category and the tails category. So the observed was 6, the expected was 5. We will square that. We will divide it by the expected, which was 5. Okay, we're going to add this to this equation for the tails category. So observed was 4, expected was 5, squared divided by 5. Okay, and that's going to be our chi-squared value. So here, let me just do some basic arithmetic here. This is going to be plus 1 over 5 squared plus minus 1 squared over 5. So 1/5 plus 1/5 equals, let's see, 2 over 5 or 0.4. Okay, so the chi-squared value is 0.4. And we don't have to take the square root of this, so you can just ignore that square. This is the actual value we're going to need to look up the probability of seeing this much deviation in an experiment. But let's wait let's put this number on the back burner and let's go and analyze the second experiment. So with the second experiment, we tossed the coin 10 times. We saw 10 heads and 0 tails. So those were the observed. We expected 5 heads, 5 tails. So we need this formula again. And use this formula for heads and for tails to add those together. So let's see, 10 for observed minus 5 expected divided by 5 plus 0 for observed minus 5 expected divided by 5. So we have some basic arithmetic here. 5 squared divided by 5, which is going to be 25 divided by 5, which is going to be 5. And this is going to be, again, minus 5 squared, 25 divided by 5, so plus 5. So chi-squared value for this is 10. Okay, so a lot bigger than 0.4. So let me summarize. So the chi-square value for experiment 1 is 0.4. For experiment 2 is 10. Now this is where we need, okay, we need one more number. We need to figure out the degree or degrees of freedom in our experiments, and this is determined with a simple formula of degrees of freedom equaling n minus 1, where n is the number of categories in the experiment. So our experiment is simple. We have 2 categories, large and small. So 2 minus 1 equals 1, and the two categories are heads and tails, right? So heads is one category, tails is the other. If we were using the chi-square to test to analyze ratios of a dihybrid cross, where we had yellow and green and round and wrinkled, that would be 4 categories. Degree of freedom would be 3. But only 2 categories in this heads and tails example. Degree of freedom is 1. Okay, so for both, degree of freedom is 1. Okay, so now what we need to do is look up. Now I have this table here somewhere. Where is it? Okay. Now you would be given a table like this on the exam. So percentage points of the chi-square distribution. So here we have all these percentages here. And we are only concerned with these chi-square values in this top row here. So the percentages are in the header column or header row. This first row right here, degrees of freedom, this is what we're interested in. So we have 2 chi-square values, right? We have 0.4 and we have 10. So we need to find the percentages to which 0.4 falls in between. So 0.4 falls between 0.102 and 0.455. So this one right here, 0.4, we're expecting the best way, let me think of the best way to put this. So with a chi-square value of 0.4, what that tells us is that we expect that amount of deviation in our experiment to occur between 75% and 50% of the time. 75% to 50%. Okay, so that seems like this amount of deviation should occur more than half of the times we do this experiment. So we would say that's not significant. That deviation is not significant. So let's go to experiment 2, where we saw 10 heads and no tails. Chi-square of 10, well, okay, that's way down here. It's off of our table. So we would expect this type of result with a chi-square value of 10. Well, we would expect to see this much deviation from the expected ratio less than 1% of the time we do that experiment. If we had a better chi-square table, it might even be less than 0.1% or 0.01% of the time. So this is highly unusual. So we're going to say this deviation is significant. And we're going to need to go look at our coin or something. It could be that, the coin was heavier on one side than the other, assuming we used a different coin for experiment 2 than we did for experiment 1. So the thing about the chi-square test, which is, kind of confusing to, to me and students, is that, we're not saying it's impossible to flip a coin 10 times and have it come up heads each time. We're just saying that it's very rare, and it's so rare it suggests something is wrong with either the experiment or our prediction. But anything is possible. So what geneticists like to do is they like to use this 0.05 value. And so usually we're pretty comfortable with deviation that is expected to occur more than 5% of the time. Not very comfortable with deviation that is expected to occur less than 5% of the time. Now let's go back to our genetic example, our imaginary genetic example, where we had 80 large peas and 20 small peas. And we were expecting a ratio of 3:1. But this is a ratio of 4:1. So we need to plug these in to this equation to perform the chi-square test, right? So one thing that's a little different is we need to figure out how many we expected. Like I told you, we expected a ratio of 3:1, but we need to figure out how do these translate into numbers of p's? So we need to look at the total number of p's, 100, and for large, we expected 3/4 of the 100 to be large. So expected 75 large p's. And for small, with a 3-to-1 ratio, what that means is we expected 1/4 of the total p's to be small. So our observed were 80 large and 20 small, and our expected, we get that by totaling these and then going to our expected ratios, figuring out the numbers. Our expected are 75 large, 25 small. So now we're ready to use this formula here. So for the large, we are going to have observed 80 minus 75 expected squared divided by 75. And for the small category, we are going to have 20 minus 25 squared divided by 25 for expected. Some basic arithmetic, we will have 25 over 75 plus 25 over 25, so 0.333 approximately, or 1/3, plus 1 equals 1.333. So that's the chi-square value. Again, you don't square root it. Degrees of freedom is 1, right? Because we have 2 categories, large and small. So 2, and the formula for that is, right, n minus 1. N is 2, n is the number of categories. 2 minus 1 is 1. So we look up 1.333 in our chi-square table here. 1.333. Okay, we see it right here. And we're expecting that much deviation from a 3-to-1 ratio in our experiment, involving 100 offspring, to occur between 25% and 10% of the time. Like I alluded to before, as long as we're expecting the deviation to occur more than 5% of the time we do the experiment, then we're happy. We're happy with the outcome. We think it supports our hypothesis. So everything is good. Now, if we had a chi-square value way down here, then we would probably need to revisit our hypothesis and start thinking about the possibility that this trait isn't caused by the homozygous recessive condition for a single gene. Well, that's essentially the chi-square test, or all we need to worry about for the chi-square test. Now, why do we even care about the chi-square test? So it might be, well, at this point in genetics, so it's nice to focus on the chi-square test because it helps us remember that these ratios are not exact, right? They're predictions, and when you do the experiment and you have the chance events involved, we don't expect the ratios to be exact, but we expect there to be small amounts of deviation from these ratios. Also helps to realize that there are random events involved during the transmission of genes from parents to offspring. And it also introduces us to a technique that is out there and that one can use to measure the amount of deviation and predict if it is unusual. So let me put those on paper. Ratios are not exact. It also helps us know or remember random events. Chance events. It's probably better. Chance events occur during gene transmission. And techniques to measure probability of observed deviation exist. So these would be 3 reasons why we cover the chi-square test at this point in genetics. Okay, so that's it for the chi-square test. And next in the course, we're going to start looking at modified Mendelian ratios.